In this tutorial, we will draw on the knowledge that we obtained from the Intro to Adaptive Immunity tutorials to understand how vaccination harnesses the power of the adaptive immune system to provide long-lasting protection to a number of childhood illnesses. So one thing I wanted to mention right off the bat is that there is profound evidence that prophylactic vaccination has a protective effect for a variety of illnesses. So this chart indicates a number of illnesses: diphtheria, measles, mumps, pertussis, polio, rubella, tetanus, Haemophilus influenza, and hepatitis B. The first column shows the peak number of cases in the United States and the year that those peak number of cases were observed. The second column indicates the peak number of cases in the year 2009, and the third column indicates the percent change. I think it's fairly obvious that these illnesses, which were somewhat common prior to the introduction of these vaccines, are now present in some cases, almost virtually eliminated, and in other cases at levels much lower than previously. And vaccines can be really important to reduce disease burden. And I've shown here a graph, now a couple of years old because time keeps marching on, but this graph shows the ability of vaccination to prevent hospitalization rates in individuals infected with COVID-19. And what this y-axis shows is the rate of hospitalization per 100,000 people. And this x-axis shows the date. And what you can see is that fully vaccinated persons have a much lower hospitalization rate than unvaccinated persons. And remarkably, despite the many changes we have seen in the COVID-19 virus, the many variants that have emerged, this relationship has really stayed the same, where vaccination has been remarkably effective at preventing hospitalization, severe disease, and death, even as it remains less effective at preventing transmission and mild to moderate disease. And the reason that this vaccine and other vaccines work to function this way is that they induce immunological memory. And immunological memory is a cardinal feature of the adaptive immune response. What I've shown here is in this dashed red line, a pathogen that is infected and over the course of a week undergoes a period of replication, but then is cleared by the immune system. The blue line indicates the number of pathogen-specific T cells over time. And as you can see, early on, we see very, very few pathogen-specific T cells, but those T cells undergo clonal expansion and eventually peak and form a large population of effector T cell responses with potent antimicrobial functions. Now, once the pathogen is cleared, you don't want all these effector T cells to remain around, and so most of these effector T cells die off. In fact, 90 to 95% of them will die off over the course of a few weeks after the pathogen is cleared. But a remnant of those effector T cells will remain as long-lived memory T cells, and these memory T cells are present at higher frequencies than the naive T cells that initiated the response. So they're at higher frequency, but also importantly, if you get a secondary challenge, they develop immediate effector functions and undergo a second round of clonal expansion. Importantly, these memory T cells can be very long-lived, up to a lifetime in some cases. And so one of the key focuses of vaccination is to induce these memory T cell responses. Multiple exposures to antigen also enhance antibody-mediated immunity, and we can get very long-lived antibody responses in this way. We learned that long-lived plasma cells can produce antibodies over a period of years so that serum immunoglobulins can persist for many years after an infection. We also learned that secondary activation of memory B cells boosts the number of long-lived plasma cells, meaning that you get increased amounts of antibodies circulating. And lastly, we learned that when memory B cells undergo secondary activation, antibodies can undergo changes to acquire increased affinity for their target. And so we not only have more antibodies after a secondary boost, but an increased affinity of those antibodies. And that's indicated in this cartoon where we see a primary, secondary, and tertiary immunization of an individual. And with each immunization, we get an increase in the serum IgG, but also an increase in the affinity of that serum IgG, leading to potent immune protection mediated by these antibodies. Okay. And one way that is kind of a good visual way to look at it, this is from a study that was done fairly early on after the introduction of the COVID-19 vaccine, looking at individuals that were vaccinated that either had pre-existing antibodies to the spike protein, meaning they had been previously infected with SARS-CoV-2, or had never been previously infected with SARS-CoV-2 and had no detectable antibodies to the spike protein. And what you can see is that in the days after vaccination, the uninfected individuals induced an antibody response, but those individuals that had been previously infected, that were referred to in this paper as convalescent, produced antibody responses that were several orders of magnitude higher than what they got from just infection alone. And so this is a really good indication of how antibody responses work. The more that you boost those antibody responses, the higher those antibody titers will get. So what kind of vaccines do we want to get? Well, we'd like vaccines that induce antibodies. Antibodies are good at neutralizing, they're good at opsonizing, they're good at activating complement, as we mentioned in the intro to adaptive immunity tutorials. We'd like to induce T cells that are killers and T cells that are helpers. We'd like these antibodies and these T cells to provide long-lived protection. We'd like them to disrupt transmission, but in the absence of disrupting transmission, we'd like them to disrupt illness. But in the absence of disrupting illness, we'd like the immune response to disrupt serious illness and death. And we'd like to be able to develop vaccines for other applications as well. So, for example, can you imagine a cancer vaccine that protects from malignancy, or an immunotherapeutic vaccine that can alter disease pathogenesis? And lastly, this is very underrated, but we really need vaccines that are safe, cheap, and stable, that can be distributed easily to all parts of the world. So what are some of the commonly used vaccine strategies to induce B cell and T cell responses? Well, one obvious way is to introduce a live pathogen. And usually when that happens, these pathogens have been attenuated such that they undergo limited replication in their host. Examples of live pathogen vaccines include measles, mumps, rubella, polio, and varicella, or the chickenpox vaccine. And these are very effective vaccines. A second approach has been to use killed vaccines, and this is due to the fact that live vaccines tend to have somewhat more adverse effects than killed vaccines, and so killed vaccines have been preferred in many cases. These are vaccines that are chemically inactivated or fixed, and because they're dead vaccines, they undergo no replication, but is a source of antigens to which the immune system can respond. And so influenza, pertussis, rabies, and polio are all examples of killed vaccines. You'll notice that I listed polio as a live vaccine and polio as a killed vaccine. That's because the oral version of the polio vaccine is live, whereas the injected version of the polio vaccine is killed. We only use the injected or killed vaccine version of polio in the United States today because it has lower adverse effects, especially for the immunocompromised, and we don't have polio cases in the US currently. The third type of vaccine that is prominent are subunit vaccines. So instead of immunizing somebody with a whole virus, instead we inject a protein from that virus or a polysaccharide derived from that pathogen. And so I've shown here a polysaccharide and a protein, and examples of subunit vaccines include vaccines to hepatitis B, Haemophilus influenza type B, meningococcus, pneumococcus, and there's many others. This also includes things like toxoid vaccines, and so these are vaccines that induce antibody responses to the toxins produced by diphtheria and tetanus. So these antibodies don't even actually bind to diphtheria and tetanus, they only bind to their toxin. And once those antibodies bind to their toxin, disease is eliminated. And lastly, I just wanted to introduce RNA vaccines because, as we all know, with the COVID-19 pandemic, RNA vaccines have now moved to the forefront of new vaccine technologies that we are very interested in. This is a ready-to-go mRNA transcript that is packaged within a lipid nanoparticle that can be then injected, fused to antigen-presenting cells, and serve as a source for protein production. So in this case, the COVID-19 vaccines produce the spike protein or the S protein of the virus to induce neutralizing antibodies. Okay. Now, one of the things that I mentioned is that it is important to induce an inflammatory response in order to enhance or in order to activate the immune response, and especially T cells to respond to a pathogen. Well, many of these vaccines, especially the inactivated vaccines or the subunit vaccines, are not on their own really good at inducing inflammatory responses. It's been known for a while that live viruses actually induce stronger inflammatory responses. But it's noted that for polio, the introduction of the inactivated vaccine really rapidly reduced polio cases over the course of several years, long before the introduction of the oral vaccine in the early 1960s. And it was noted that early on, dirty batches of the killed polio vaccine worked better. And it was later discovered that this was probably due to contaminating bacterial products within those batches that enhance the inflammatory response and therefore enhance the immune response in those individuals. And this has led to the idea of using a class of molecules called adjuvants. Adjuvants are chemical additions to vaccines that induce inflammation, and they're especially important for inactivated, subunit, or conjugate vaccines. So adjuvants induce the activation of macrophages, inflammatory cytokine production, and all the inflammatory response subsequent to that. Again, they're important for non-live vaccines, but unfortunately, the mechanisms underlying adjuvants are really poorly understood. The most commonly used adjuvant in humans is alum, but this is an area of active investigation, and there are a number of new adjuvants that will be on the market or have entered the market already. Now, I've shown here the schedule for immunizations for children and adolescents up to 18 years. This is just for your information and perusal. I'm not in any way, shape, or form expecting you to memorize this schedule, but I think it would be of interest to you to familiarize yourself with it, especially as we discuss the impact of childhood vaccination on preventing disease. You can access this schedule at this link indicated in the bottom of the slide. And we do have a number of new challenges for new vaccines and a number of challenges to think about in how we apply the vaccines that we already have. For example, is route important? Is it matter if a vaccine is, for example, injected versus whether that vaccine is inhaled, especially if it's a vaccine for a respiratory virus? And an example of an inhaled vaccine is the flu mist vaccine. And there's still a lot of work to be done to understand how the route impacts protection mediated by the vaccine. The timing can be very important. Do we administer vaccines immediately after birth? Do we wait a few months, or do we wait a few years? There are some vaccines that are effective when administered right after birth, including the hepatitis B vaccine, but other vaccines for which we wait quite a while, including the measles, mumps, and rubella vaccine, which are typically administered only after 6 months to a year. We have a lot to learn about dosing schedules. How many boosts are ideal? How far apart should those boosts be to maximize the efficacy of the boost? And then we need to develop vaccines for organisms that are highly polymorphic, that have variable expression of surface proteins. Malaria is a good example of this. We need better vaccines for pathogens that are really good at developing escape mutations. Influenza, HIV, SARS, those are all highly mutable pathogens, and HIV has been particularly difficult for vaccinologists. And there are many cases in which vaccine distribution is abysmal, and that includes the vaccine for measles, which is highly effective, but is not distributed effectively to many parts of the world, leading to numerous cases of measles deaths every year worldwide. So that is the end of our vaccination lecture, and feel free to ask me any questions that you may have based on this material.